Chapter 4 is going to get us through taxonomy, and taxonomy and bacteria, and the archaeans for that matter, is still honestly in a state of flux. If you were to open up 3 different textbooks about bacteria, you would find 3 different taxonomic systems to describe those things. So I want you to keep that in mind. I'm going to use the one that your textbook uses. I do have sort of an issue with the way your textbook jumps around between different taxa, but I did create a summary slide to try to show you what they were attempting to do. I just don't love that they sort of jump around back and forth between things because it can get confusing as you look through that. With all of that said, we'll go ahead and talk about some of the things that need to be talked about, although a lot of this chapter is just going to be straight up memorization, and I recommend you make some flashcards to help you study this stuff. So to start, I always want to talk about symbiosis because people tend to come out of high school thinking that symbiosis and mutualistic relationships are the same thing. Mutualism is a type of symbiotic relationship, but so is predation, and so is herbivory. All of those are symbiotic relationships. So symbiotic just means you have things that are living together and interacting with each other. Mutualism means they're living and interacting and everybody wins. Everybody's happy in a mutualistic relationship. That's not the only type of symbiosis. I just really wanted to make sure you understand those terms aren't synonyms. Mutualistic relationship is a type of symbiotic relationship, but so is commensalism and parasitism and again predation and things like that. They all are types of symbiotic relationships. Okay, I wanted to take a moment to talk about the microbiome just a little bit. Your book spends a pretty decent amount of time talking about it here in this chapter. Your microbiome is very important for your overall health and well-being, and there is a ton of research that is happening right now in relation to the microbiome, partly because of the microbiome project that your book discusses at one point. We are learning a lot of different things. Different cultures have different microbiomes. One of the things that is happening in terms of research is they're trying to go back and find, like, what is the ancestral microbiome for a typical hunter-gatherer, and how has that changed over time? Are Americans getting more obese because of our microbiome or because of the food that we eat? Did our microbiome change because of our food, or did it change and then our food changed? There's just a lot of things that we don't know that's associated with that. Some things to stress: your microbiome is different in different places. You have different species living in your nose than you do in your lungs, than you do in your bladder. A lot of times, especially in nursing and med school, they talk about organs being sterile. There's really not a sterile surface anywhere inside of your body. There are microbes literally everywhere, including within your cranial vault where your brain is. It's just that whenever we stick medical instruments into those places, we need those to be sterile so that we don't introduce new species into those places. But yeah, even if you take a blood sample, there's going to be bacteria in that blood sample. You just literally have bacteria living everywhere inside of you. Just different things in different places. One of the things that can be very harmful is if you get a species that's adapted for your nose and then it gets into your throat, well, in your throat, it can cause problems. And so now you have a disease that is considered opportunistic because that species doesn't normally cause disease, but because it moved to a new place, now it is. That happens all the time, especially in relation to a hygiene issue. When a person, this is going to get gross really fast, but when a person is wiping after they have gone to the bathroom, a lot of people don't understand that they are not supposed to wipe from back to front. And when they do that, they smear feces along with all of the microbes that are in that feces up towards the urethra. And that allows the bacteria that normally live in your colon to get into your urinary tract, and it is not adapted for your urinary tract, and your urinary tract is not adapted for it. And so that's when you end up with urinary tract infections. And so you want your microbiome to be there, absolutely, but you want it to stay where it's supposed to stay and not move into new places. All right, from there, your book gets into the taxonomy and it starts hitting different groups fairly hard. At first it talks about, well, there's a couple of groups of gram-negatives, and then it's like, well, there's 4 groups, and then it's 5 groups, and it depends on what level you're talking about. There's different numbers of phyla than there are classes and there are orders. The ones that I have up here for you right now, and honestly, it doesn't even mention the cyanobacteria, and they should be in there. So I really should have a number 5, but for some reason I didn't. Your book, at this point, only mentions the top 3, the proteobacteria, the CFB group, and the spirochetes. Later on, it mentions planktomycetes, and then after planktomycetes, it also mentions cyanobacteria. Those are all phyla within the kingdom bacteria, which appears to be the only kingdom in the domain bacteria. This happens to be a GIF of a spirochete, and you're just seeing, so this is this particular phylum, which is number 3 up there. So this kind of goes back to your book gets really annoying because it sits there and says, well, there's 3 groups of gram-negatives, but then it goes on and describes 5 groups. So again, I've got a summary chart at the end, and hopefully that'll make things a little bit easier for you to understand. It does the same thing here. It's like, hey, look, there's 2 groups of gram-positives, and then later on, no, there's way more groups of gram-positives. So we're going to elaborate on those when I hit the summary here in just a second. Anytime we try to edit the taxonomic rankings, we create a phylogenetic tree, usually using some type of nucleotide sequence at this point. This is a representative phylogenetic tree for the Proteobacteria, which is a gram-negative group. What I want to point out on this slide, and I'm going to mention again on the next slide, is, so you look at all of these species that are up here, and remember, when you've got 2 species that share a branch, that means they are pretty closely related to each other. The closer the branches, the more closely related. The farther apart the branches are, the more distantly related the species are. P.S. Candidatus means this is a maybe species. It's still under review, so that's not a real species at this point. It's a candidate species, if you will. But notice chlamydia is off down here, and it's not related to any of these other guys. It's off on its own little branch off by itself. Most textbooks have considered chlamydia to be its own separate phylum, which means it's not even in the Proteobacteria group. It's in its own separate group. I tend to like it to be in its own separate group, but that's just because that's what I was taught, and I'm old, and it's hard for me to kind of get out of my rut, if you will. Your book says that it's not only not its own phylum, it's in the Proteobacteria phylum, and then it's in the alpha Proteobacteria class. So they are nesting it in here with the Rickettsia group, and it talks about Rickettsia rickettsii, which is what causes Rocky Mountain spotted fever. But what I want to show you is it's not at all related to those species. It's way off over here on its own separate branch. So personally, when I look at the trees, I see chlamydia as its own separate phylum. But like I mentioned on one of the slides, the next slide, in fact, I really don't have a horse in the race. I don't care. I could care less about taxonomy. I teach it to you because I'm required to teach it to you, but it's been in flux for like 30 years at this point. And I don't like making people memorize something that's going to change because of some new piece of evidence that we have, and yet that's science in a nutshell. So there you go. You guys think for yourself, and where do you think chlamydia is supposed to go? Here's a different phylogenetic tree. This one is unrooted. What I want you to notice is the chlamydia group is right here. Well, here's the other Proteobacteria, and again, really could not be too much farther apart. And so I kind of feel like chlamydia should be in its own separate group, but your book puts it in the alpha Proteobacteria, and if that's where you want to put it, fine. Put it wherever it makes you happy. So we spend a fair amount of time talking about the Proteobacteria because that's a very big, diverse group of bacteria. It then starts to talk about these 3 groupings, the CFB group, the spirochetes, and the Planktomyces group, or Planktomycetes group. Cyanobacteria are another phylum that it once again talks about separately, like it's its own separate thing. It's a gram-negative. It really should be listed here as a non-Proteobacteria gram-negative, but your book doesn't do that for whatever reason, and so I kind of didn't either. What I wanted to mention with the Methanobrevibacter oralis is the correlation does not equal causation thing, and hopefully you got that in your freshman biology courses. A researcher had been looking at periodontal disease, which it happens when a whole host of different factors end up making the gums recede from the teeth, and it creates pockets that allow even more bacteria and archaeans for that matter to move into those pockets and then start to ferment and produce more acid and produce more tooth rot that gets into the teeth and the bone and causes major problems. Well, after this researcher was looking at what grows in the pocket between the gums and the tooth, he found this archaean species that was in there, and so he sort of jumped the gun and said, hey, I found an archaean that causes human disease. He did it because if it were true, he would be the first person to prove that an archaean actually caused human disease. But what ended up coming out of more research is, Yeah, it's correlated with periodontal disease, but it doesn't seem to cause it. It seems to actually result from it. So the periodontal disease creates that pocket, which is just more habitat for things to move into, and so then it moves into that habitat. So it didn't cause the gum disease. The gum disease caused the habitat that allowed the archaean to move in. Appears to be where this is at this point. So correlation does not equal causation. Okay. So here's the summary of sort of everything, and I organized it because I, for better or worse, have OCD, and again, I hated how your book sort of jumped around. So there's 2 domains that are discussed in this chapter. The domain bacteria, which again, prokaryotic, uses peptidoglycan in the cell wall, uses the ester leakages, all of that wonderful stuff that we've already talked about in previous chapters. It seems like there's only one kingdom in this group. I kind of wish, you know, there was like a gram-positive group and a gram-negative group and then a we don't have a cell wall group within the kingdoms, but that's not the way it works out. There's just one kingdom in that domain. These are the phyla next that are in there. And I put chlamydia here sort of at the phylum ranking because, again, that's kind of where I think it goes, although your book puts it up here and under the proteobacteria in the class alpha proteobacteria. I give you model organisms to look at for each one of these groups. You can write them, like print this page out and then write the model organisms beside that. And again, I recommend you make flashcards with like the name of the class and then the characteristics, the name of the model organism, and then what class does it belong in. This class, and then on the flip side, what phylum does it belong in, which that would be really easy because it's in the name of the class, quite frankly. But this just sort of gets you organized. Just remember, in terms of taxonomic hierarchy, it's domain, kingdom, phylum, class, order, family, genus, species. The model organisms take you all the way down to species. Every now and then your book does take you down to the genus level for certain things. But most of the time they stay around within the class and the phylum grouping. There are just a couple of times where they dive a little bit deeper into that. So I hope that this helps you see how everything is actually related to each other. So everything from the deeply rooted bacteria through the more modern E. coli up here, those are all in the same kingdom, apparently, and domain. They are in separate phyla, though, and so that's where things start to get different. I do also want to mention, your book doesn't talk about phylum Firmicutes, but it does mention these classes. I just wanted you to know what phylum they belong in because they're not related to these others. They're in their own separate phylum. And then same here, they talk about the genus Mycoplasma, but they do not talk about class Mollicutes, which is in the phylum Firmicutes. And so I wanted to go ahead and include that one in there for you guys as well. I hope that helps you sort of see how things are working here in this chapter. Like I said earlier, the best way to study this material is probably going to be flashcards for you. So make yourself a Quizlet or go old school and buy yourself some index cards, make some flashcards and study them. It's a difficult chunk of material, but it's not impossible. You just need to spend some time studying it.